Welcome to Beaver Science. Today we're going to do a drawing of a diagram that illustrates the molecular structure of the DNA molecule described by Watson and Crick nearly 70 years ago. Okay, the drawing that we will be doing today corresponds to figure 13.8 in your textbook. So if you have your textbook, you might locate that figure and refer back to it as you have time. The drawing will require a full sheet of paper, and we'll do this in portrait style. And so either on a piece of typing paper or in your notes somewhere, label this drawing DNA structure at the top. And then as a reference, so if you want to come back to it, this is essentially figure 13.8 in your text. I'm going to start by working on the left-hand side or the left margin of the paper. And basically, remember, DNA is a double-stranded molecule, and it's kind of these repetitive sequences on the outside of the molecule, these repetitive sequences of phosphates and sugars. So just start with a P with a circle around it, and then we'll show the bond to the sugar. The sugar molecule is a 5-carbon sugar, so try to show like 5 points to the sugar molecule. And then it's just phosphates and sugars, which are the kind of outside supporting portion of the molecule. There are a total of 4 nitrogen bases found in the DNA molecule. So this doesn't have to be real long or convoluted. Just 4 sugars, which will correspond to the 4 nitrogen bases that we find in the DNA molecule. So that's one side, or the supporting portion of the DNA molecule. And now one of the things that you'll notice on your figure, and that we'll come back and talk about later when we talk about DNA replication and protein synthesis, is the orientation of the molecule. Okay, DNA is what they call anti-parallel. And so one side, it's like, 2 lanes of traffic going in opposite directions. And so one side runs this direction, and the other side runs this direction. And you'll see this business of 5' and 3', and you'll hear me refer back to that from time to time. So this side of the molecule is oriented in this direction, running from 5' to 3' this way. Well, what does that mean? That means basically it's describing the point of attachment of the phosphate to the which carbon is located on the sugar molecule. So if you look at this real quick, this is the first carbon, this is the second carbon, this is the third carbon. Each one of these points represents a carbon. There's a 4th carbon, and then here is the 5th carbon. There's another carbon right here on this bond. So this phosphate is attached to the 5th carbon on the top side of this strand of the molecule. And then if you look down, this phosphate is attached to the 3rd carbon. So you have 5' to 3' running in this direction. The other side of the molecule is running the opposite direction. So that's the supporting structure of the DNA molecule. Let's go ahead and draw in the nitrogen bases. That's where the information is stored. So everywhere you have a sugar, you'll need a nitrogen base. The first one is thymine. The next one that we're going to look at is guanine. Guanine is a little bit larger in that it has this additional carbon ring associated with it. And we'll look and talk about that here in just a minute. Then the Another nitrogenous base that you'll find in DNA is cytosine. And then we come back, and the last one is adenine. And adenine is another one that has this additional carbon ring associated with it. So you can see that we have a couple of larger molecules and a couple of smaller molecules, and they have a definitive way of combining. They combine with their complementary nitrogen base pair. Thymine always combines with adenine. And remember, adenine has that extra carbon ring associated with it. These two little bars represent hydrogen bonds, and they're important, and the number of hydrogen bonds is important. Between adenine and thymine, there's always 2 hydrogen bonds. Okay. Guanine pairs with cytosine. And so same way down here, cytosine and guanine. But remember, you have that extra carbon ring associated with adenine and guanine. And then down here, 2 hydrogen bonds between adenine and thymine. So everywhere you have a nitrogen base, we're going to draw we're drawing the complementary strand here. So now we need our sugars and our phosphates. But be careful and slow down a little bit here. On the sugars on this side, remember that the molecule is running in the opposite direction, and so it's still a 5-carbon sugar, but those sugars are going to be oriented in the opposite direction, kind of like a little upside down house. Still the same sugar, deoxyribose, but just facing the opposite direction. Now on this side of the molecule, and I'll show you this again here in just a minute, remember that 5' to 3' business. And so on this side of the molecule, on the bottom side of this strand, this would be the 5' side, and then on the top side, that's represented by the 3' side. So this side of the molecule is running from 5' to 3' in this direction. All right, let's go in and look at this just a minute on this a little bit closer. Oh, got to add our phosphates. And so from here you have a phosphate here, a phosphate here, phosphate here, phosphate here, and then the last phosphate coming off down here. A little crowded over here, but hopefully you can see that. I'll show you a cleaner diagram right at the end. So let's look at this side. This is a 5' side here, but remember the sugars are oriented kind of in the opposite direction. And so this would be our first carbon, second carbon, third carbon, fourth carbon, and then remember that there's a 5th carbon right there. And so this is running 5' to 3' in this direction. All right. One of the things about these hydrogen bonds that has an influence or an impact on the shape of the molecule is, notice that you have the 2 hydrogen bonds between adenine and thymine, 3 between cytosine and guanine. That exerts an unequal pull on the molecule, which causes the molecule basically to coil or twist. And so it's the unequal pull between the hydrogen bonds that causes the molecule to twist or coil into that, what you've all heard, double helix. So helix means twisted or coiled. That's caused by the unequal pressure of the number of the hydrogen bonds between the nitrogen bases. Let's go ahead and label these structures. So those are our hydrogen bonds. This is a nitrogen base. This particular one happens to be thymine, which indicates that this is DNA. We'll talk about that later, but RNA would not have thymine, it has uracil. Uracil replaces thymine in RNA. And then this is our sugar. This particular sugar, since we're talking about DNA, is called deoxyribose. And what that means is that this sugar has one less oxygen atom than the sugar that you find in RNA. And then out here, this, of course, is our phosphate group. And it is a PO4 with kind of a big -3 charge associated with it. DNA is a polymer, and essentially that means it's kind of a repetitive sequence of smaller molecules. And so the structural unit for both DNA and RNA is what they call a nucleotide. And a nucleotide, you can use kind of a dotted line to represent what a nucleotide is. You'll hear this term and see it again when we talk about both DNA and RNA structure. But a nucleotide includes a combination of a phosphate, a sugar, and a nitrogen base. So those three things make up a nucleotide. And that's true of both DNA and RNA. So this is a nucleotide, this is a nucleotide, this is a nucleotide, and it's just those repetitive sequences of nucleotides kind of combined together that make up the DNA and RNA molecule. Let's look real closely or quickly at the what we call complementary nitrogenous bases, and there's two major groups. There's purines and pyrimidines. And purines always combine with pyrimidines. Now I remember that around here we do farming, and another word for farming is ag. And so I know that ag is big here. And so I remember that the two purines are adenine and guanine. Those are the nitrogen bases that have the additional carbon rings. So the purines are adenine and guanine, and that makes the pyrimidines, they're complementary nitrogenous bases. Adenine always combines with thymine, and guanine always combines with cytosine. There are 2 hydrogen bonds between adenine and thymine, 3 between cytosine and guanine. Last thing to kind of keep in mind as we move forward, there is another pyrimidine, but it's in RNA. And so in RNA, thymine is replaced by uracil. And so that is basically the DNA structure. I'm going to show you just really quickly to finish up on, so maybe you can get a screen capture, a little bit cleaner graphic that I drew earlier. Same graphic essentially, but maybe you can zoom in on this and see it a little bit more detail. Thanks for joining.